Thank you, Sebastian, and welcome everyone, and thank you for joining this product brief that is an overview of the Phoenix platform. And I'm the product manager for the pharmacokinetics software offered by Certara. That does include the Phoenix platform, and the components of it that I'd like to talk about today are WinNonlin, NLME, and the validation suite. So I'll begin with talking about what is Phoenix. So Certara's Phoenix platform is the leading PK/PD modeling and simulation software used worldwide by over 6,000 researchers at biopharmaceutical companies, academic institutions, and global regulatory agencies such as 11 divisions of the US FDA. Phoenix extends the winning formula of WinNonlin by combining its trusted algorithms with built-in, easy-to-use graphical user interface and a textual editor for advanced analysis. Phoenix 8.3 offers features that support compliance and efficiency, including visual workflows that can be used and reused, locked to maintain compliance, and these workflows increase efficiency and also are able to produce reports. It produces high-quality tables and figures, and is also compatible with CDISC. So if you have CDISC SDTM for clinical or send data for non-clinical, that type of data preparation can be done in the Phoenix platform. And Phoenix has a project-centric design to easily store, share, and reuse Phoenix projects. Phoenix also has a fully integrated validation suite for fast automated software validation in under 30 minutes. So conceptually, what Phoenix is an operating platform. It's got a single user interface with a look and feel. So the single user interface has several different modules to it. It includes WinNonlin, which is used to perform individual PK/PD modeling, non-compartmental analysis, and bioequivalence analysis. The IVIVC toolkit, which is used to do in vitro, in vivo correlation analysis that is used in formulation development. Phoenix also has an NLME, which stands for nonlinear mixed effects module, which is used to perform population PK/PD analyses. The Phoenix platform also has a validation suite that's used to perform fast automated validation for WinNonlin and or NLME. Additional plugins that are available include CDISC and the PK Submit plugin, which is used to get non-compartmental analysis results into a CDISC compliant format. Lastly, we have a secure central data repository called Integral, which Phoenix can connect to pull in data for analysis, run the analysis, and then save back in a manner that is used to achieve 21 CFR Part 11 compliance. So all of these different modules are in a unified user interface that has the same look and feel. So it's very easy to go between the different modules. The Phoenix platform has a large array of useful tools that span the PK/PD analysis spectrum. These include system tools, such as a workflow. A workflow is a series of steps that one uses to go from raw data, prep data for analysis, run modeling and simulation, and then tabulate results. That series of steps is a workflow. And it also supports data links, so you could make a template of your workflow and pipe new data in and run it through the same workflow steps for the types of studies that you would repeat often. There are also a variety of data prep tools, such as CDISC data preparation and various spreadsheet operations, such as append worksheets, cross-product worksheets, data wizards for doing transformations, merging and joining worksheets, pivoting worksheets, and so on. There are also a variety of plotting tools available in Phoenix, and these include typical scientific plots such as bar plots, column plots, histograms, XY scatter plots, matrix, and categorical XY. Okay, some additional tools that Phoenix has from the analysis perspective. Probably the most commonly used tool in the Phoenix platform is the non-compartmental analysis tool. It's used very commonly in preclinical or clinical studies, but primarily preclinical. We also have a bioequivalence analysis tool and several other analysis tools, descriptive stats, non-parametric superposition, and so on. There are also a series of compartmental PK/PD modeling tools. So they include PK and PD compartment models, PK/PD link models, Michaelis-Menten models, linear models, and indirect response models. One can also do dissolution models for formulation development. We also have a very flexible Phoenix model. The Phoenix model is a maximum likelihood modeling engine that can be used to perform either individual or population PK/PD analyses. And there's a very handy model comparer tool, which can be used to compare objective functions and other diagnostic outputs from various models that will help guide model selection. Okay. Phoenix also has some reporting tools. It has a table module that allows one to construct a report table, tabulating results in a very easy to read format. Different layouts are supported, including summary statistics. And there's also a reporter tool, which one can use to selectively pull outputs from your Phoenix project and assemble them into a presentation quality report, complete with figuring and legending and captions and footnotes and the like. The Phoenix platform can also be used to integrate with third-party softwares such as NonMem, SAS, SigmaPlot, PearlSpeak's NonMem, and R. A relatively new feature in the Phoenix platform is the ratios and differences tool. So very commonly in these types of studies, in various studies, one might want to compare PK parameters from different routes of administration or from different dosing regimens, parent metabolite, and so on. So this ratios and differences tool was added to very easily support studies such as average renal clearance when you have plasma and urine data, accumulation ratios for multiple dosing studies, demographic comparisons such as gender differences, parent to metabolite exposure ratios, dose proportionality, as well as exposure ratios for topics such as food effect, drug-drug interactions, and relative bioavailability. So the user interface for Phoenix, the diagram here is essentially showing a workflow that allows you to visualize the series of steps that go from raw data all the way through analysis and to the final result. So as an example here, you might have an input data set that pipes in through a data link, maybe do some exploratory plots, such as a concentration versus time plot by subject and analyte, maybe make a table of individual concentrations and mean concentrations with descriptive stats. From there, after doing that exploratory work, perhaps run a non-compartmental analysis to compute PK parameters, and then make a summary of those parameters in the form of either descriptive stats or a table. So this series of steps allows one in a workflow to get from the raw data all the way through to the endpoints. Okay, so that's the Phoenix platform. What is WinNonlin? So WinNonlin is the industry standard for non-compartmental analysis and PK/PD modeling and simulation. Phoenix WinNonlin has been around for over 30 years and is used by thousands of scientists in over 60 countries, including top pharmaceutical companies and academic institutes. And we also have Phoenix made available to several regulatory agencies around the world, including the US FDA, Japan's PMDA, the China Food and Drug Administration, and UK Medicines and Healthcare Products Regulatory Agency. And all of these organizations have Phoenix WinNonlin available to evaluate drug submissions. So WinNonlin, the WinNonlin component of Phoenix, supports non-compartmental analysis and individual PK/PD modeling, as well as a variety of statistical analysis tools for a wide range of studies and analyses, as I alluded to in the previous slide. The NCA engine automatically outputs additional parameters for plasma and urine to save time, reduce errors, and produce higher transparency for regulatory agencies. It is possible to set strict criteria for NCA, the acceptability of results. We call those business rules, as well as to compute user-defined parameters for the NCA. It's also very easy to calculate the NCA parameter ratios, which increases efficiency and saves time. For early non-clinical research to large clinical trials, the NCA engine and statistical analysis tools can be used in single, multiple, or steady state dosing studies, drug concentration in plasma or urine, sparse sampling studies, as well as toxicokinetic studies, pharmacodynamics data, formulation comparison, first in human dosing, single and multiple ascending dose, drug-drug interactions, and bioequivalence studies. I just wanted to point out, again, we do have collaboration with regulatory agencies such as FDA that allows the FDA to provide input and work with us to optimize the software. So one of those types of collaborations, this is a poster from AAPS. And so we worked with the FDA to create a workflow template that performs reference scaled average bioequivalence. And we have templates for the progesterone guidance. That's kind of the baseline reference scaled average bioequivalence. But typically these reference scaling is done when the drug has high intra-subject variability. So it's important to be able to scale the confidence limits and adapt the standard confidence intervals for bioequivalence. And this particular example I'm showing was from the warfarin narrow therapeutic index drug study. And it shows how we were able to make a workflow template to satisfy all the criteria from the guidance. Okay. As far as Phoenix WinNonlin is concerned, just to get an idea of how often we release new versions. So Phoenix first appeared in June of 2009, and we typically release a new point version about every 1 to 1.5 years. In the Phoenix 8.3 series, we did a series of maintenance releases, but we're also working toward a Phoenix 8.4 release. So it is an active live software that is continuing to be improved and developed over time. I did want to allude to the fact that Certara does take a proactive approach to engage our customer base for ideas about product improvements and enhancements. These include live surveys during webinar series, user group meetings, client visits, training courses, as well as user forums. A summary of the key features in the WinNonlin 8 series will be shown in the software demonstration. Okay. So with that, I would like to go ahead and pause the slides here and move into the Phoenix application and have a look at it. Okay. So what I'm showing here is the Phoenix application. I'm going to show an example project. I'll start with the data set. So in this data set, we have a series of PK data in an input spreadsheet. And this spreadsheet has all of the data of a given type in a column. So all of the subject IDs are in one column. All of the formulation names, such as Tapsler or Cablet, are in a second column. The times of the observations are in a third column. And then the concentrations in nanograms per milliliter are reported in a fourth column. So just to show, kind of an example workflow, what one might do is send this data set to an exploratory plot. Perhaps plot the individual subjects and group by formulation. So for subject 1, if this is a crossover study where each subject got 2 different formulations, you can see the capsule data and the tablet data plotted for subject 1. and then toggle between the different subjects on different pages. A second plot would be to overlay by formulation. So in this case, on each page, we have a different formulation. So page 1 would be the capsule data for all subjects, and page 2 would be the tablet data for all subjects. And these are plotted on a semi-log scale with the log of the concentration on the y-axis and the time on the x-axis. From there, the user could go to the non-compartmental analysis tool, and every tool has a setup area. So in the setup area, that's where you define what data type each column is. So you can map the time and concentration columns and the profiling columns, such as subject and formulation, that will run a separate NCA analysis for each combination of subject and formulation. There's also an easy-to-use dosing input where you can enter the dosing information for each subject. There's also a graphical slope selector that can be used to select the terminal slope for each individual profile. There is a default best fit algorithm, but it's also possible to specify a time range for the terminal phase. So those are some key features. A couple other items here is that you can set, it's good at unit handling. So you can take units from the input data set and maybe convert them to some preferred units for your output table if desired. Let's say you wanted concentrations in micrograms instead of nanograms, or doses in milligrams instead of nanograms. It's very straightforward to do that here. You can also see the parameter name list. This is a list of all of the output parameters that come from NCA. And you can, they have default names, but you can also specify your own preferred names in that, as well as toggle whether or not to include each parameter in the workbook result. Once the non-compartmental analysis is run, you get several outputs. I'll start with the plotting output. The plot will show on a profile by profile basis the terminal phase, which is done by a log linear regression of the points toward the end of the profile. That's represented by the green regression line shown on the plot. And that's used to compute parameters such as adjusted R-squared, as well as the half-life. In the final parameters pivoted output, you get a table that shows a listing from left to right of the subject, formulation, and then associated PK parameters. So I alluded to the half-life here. You also get the typical parameters such as Cmax, AUC, AUC infinity, AUC percent extrapolated, volume of distribution, clearance, and so on. So it's very easy to take this result worksheet and then put it into other items such as a table. So if I wanted to tabulate and summarize key PK parameters from this study, I would set up a table. The table does have a setup interface that allows you to construct different layouts for how you want to present the data. They do include precision, so you can toggle between, say, for example, significant digits and or decimal places, and how many of each you want, as well as the ability to include summary statistics in an easy-to-use checklist for inclusion in the table. Once you run the table, in this result, I set it up so that there's a page break on the formulations. So this first table toward the top is all about the capsule formulation. There's a vertical listing of the subject IDs, and then the key PK parameters Cmax, Tmax, half-life, AUC last, and AUC infinity. Notice that these are formatted to 3 significant digits for the AUC and concentrations. For the time parameters, they're formatted to 2 decimal places. And then the summary statistics are shown in bold below each column. And then the same layout is also used for the tablet formulation. So the exact same formatting. You can also do advanced formatting such as superscripting and subscripting parts of the name, using different symbols such as the Greek alphabet and the infinity symbol, and so on. From there, if you have enough results in your report, in your project that you may want to report them, you can send the results to the reporter tool. So the reporter tool has an interface that allows you to pick selected outputs. So maybe this is the grouping of exploratory plots, This is the group, a second set of exploratory plots. And again, this is just the setup preview. It's not the actual report. But you can also do things like take your lambda z result plots, final parameter table, as well as the table that was constructed earlier in the workflow. And you have also different options for captioning using major and minor indices to give figure numbers, and toggle whether the output format is in Word, PDF, or both. And also identify whether you want to include footnotes, such as the date timestamp of when the analysis was run, what is the source, and the object name. But once you run the table, you can get the results in either a Word format, such as here. So this is an example in the Microsoft Word format. It shows all of the exploratory plots in both linear and log format in a lattice. You can also look at the additional outputs, such as the final parameters listing, all the way down to the summary tables at the bottom. And notice that the effect of the page break, it tabled them out into table 1.2 for the capsule and table 1.3 for the tablet. So that's the Word format, and you can also get it in PDF format as well. It's kind of nice and easy to navigate as well in the PDF format. So whichever format you prefer, I can jump to the end here for the PDF. This reporting tool is part of the Phoenix platform. So that's just an example workflow. I did want to introduce also the topic of ratios and differences. So finishing up with this particular project, I'm going to go to the non-compartmental analysis and just show some basic items that were set up here. So on this particular NCA, I set up a rule that required that for final parameters to be accepted, they must have an adjusted R-square value of greater than or equal to 0.98, and that the AUC percent extrapolated must be less than or equal to 20%. And so what ends up happening when you set up these business rules is you get these flags in the output. So for the R-squared adjusted, we see an example here. Here's R-squared adjusted. It will go through and it will find any, for example, this one is less than 0.98, and it doesn't filter it out, but what it does is it makes a flag column that says whether it's accepted or not accepted per the business rule criteria. And from there, you can use that flag column to, if you want to filter out those parameters, you can use a data wizard as an example to filter, basically essentially filter where the flag is not accepted. And then in the results, you see which records were filtered out, all those that were not accepted are filtered out, and all those that were accepted remain in the results. So that's just a base example of some business rules. I did want to show a further example of that. So with these business rules here, you can do things like LLQ replacements. So that's done via this BQL tool, where you can identify when do you replace the LLQs. So this is the original values. And early in the profile, you might want to replace BQL with 0. Later in the profile, you might want to replace it with missing, as an example. So those replacements can be done. And then in the business rules of the NCA, you can specify things like the rules including span. If the span parameter is the number of half-lives, so this business rule is saying the profile must have a span of at least 1 half-life in order to be valid. And then the data tools can also be used to support advanced reporting tables, such as this is an example of PK parameters with stats in the body. So if you want to show the mean Cmax plus or minus, the standard deviation or other PK parameters, these types of tables can be composed with a combination of data tools and the reporting tool. Here's one other example. If you get the table and you want mean concentrations instead of PK parameters, this is a listing of the actual concentrations by nominal time, with reporting the concentrations for capsule and tablet at each time point, with the mean plus or minus standard deviation with the CV percent reported. So advanced tables can be constructed. And then I'd like to show one more example here, and that is going to be the ratios and differences tool that I alluded to in the slides. So in this case, we have a large data set. This is from a toxicokinetic study, and there are different routes of administration, different matrices, gender information such as females and males, different routes of administration, as well as different analytes such as parent and metabolite. This might be test analyte, and then 4-hydroxy-TA is the metabolite. And so that supports different ratios such as dose proportionality. So there's different dose levels. There's a 1 mg per kg, 10 mg per kg, and 60 mg per kg. So you might want to look at dose proportionality and see if the scaling is linear across dose groups. And then you might want to look at the result and get these ratios. So maybe the Cmax of the 10 milligram compared to 1, the Cmax ratio of the 60 compared to 1, and same ratios for AUCs as well. You can also look at things such as accumulation ratio. So in this case, we're comparing day 14 to day 1. So there's a day column here that identifies the day, and then you can set up the ratio in this bottom panel and come up with ratios, accumulation ratios comparing day 14 to day 1. And you can also very easily do these for gender differences. So that would be this unstacked gender differences, Cmax ratio, female to male, AUC last, female to male, as well as parent to metabolite ratios. So if you wanted to compare parameters, parent to metabolite, you can very easily set these up as well. So this is a very handy tool for different types of study types. The core of this is the NCA, or non-compartmental analysis. And from there, a variety of easy to use data tools can really help you get the most out of your NCA. Okay, so that's Phoenix WinNonlin. At this point, I'd like to switch back and introduce the topic of NLME. So this is another module in Phoenix. NLME stands for Nonlinear Mixed Effects Modeling. NLME is used to perform population PK/PD analysis, and the aim is to expand a PK/PD model to account for sources of variability in key model parameters between subjects in a population. These are things, the questions that can be answered by POP modeling include things like, does a drug's clearance increase with the age of the subject? Does volume of distribution vary with body weight? Is there a significant difference between sexes for any important model parameters, and so on. So a good population PK/PD model will not only describe PK/PD outcomes in the population being studied, but will identify key demographics, also called covariates, that can be used to produce PK/PD outcomes in a different population of subjects. So study types where one might use NLME include therapeutic drug monitoring, pharmacometrics, PK/PD and PK/PD link models, physiology-based PK models, also known as PBPK, disease progression models, and they're also an important prerequisite for clinical trial simulations. So some key features of Phoenix NLME include different modeling algorithms. So that, kind of the base or simplest algorithm that one might use early on is the naive pooled, where you can do individual or pooled data analysis. And then there's also modeling engines that include parametric methods such as first order conditional, Lindstrom-Bates, and extended least squares, iterative two-stage Laplacian, as well as QRPEM. The QRPEM is a PEM method, and it's a very advanced method that won a modeling algorithm contest at Page in 2012. So QRPEM was found to be consistently the fastest to converge among the expectation maximization, or EM methods, demonstrating the advantage of this approach. Also, in population analysis, stages to it. So first, model fitting, developing a structural model as well as a residual error model. That can be done in the simple mode. And then when one goes to find covariates that may be significant, there are covariate search methods. They include Okay, apologies for the delay there. I lost my Wi-Fi briefly, but I'm back now. So I was showing the key features. So I'll start on this bottom portion. When you're doing population model fitting, the simple run mode can do things like help you build the structural model and identify the best structural model, as well as the residual error model. There are covariate search methods available, such as the stepwise approach that can be conducted in either the forward or backward direction. Shotgun approach, if you have a large number of covariates and you just want to kind of look at large numbers of combinations of them, that's available, as well as scenarios where you handpick different covariates and include them in a model fitting scenario. When it comes to model validation, the standard methods are available. One can do bootstrapping, profiling, as well as visual predictive checks. And it's also possible to take the population PK/PD modeling results and run simulations based off of them. Okay, so that's the Phoenix NLME. I'd like to go back into the Phoenix software and go ahead and show that feature. So this is a population PK/PD analysis conducted based on Phenobarbital in infants. So we start with an exploratory data analysis. So this is the PK data for all of the subjects. From there, one might perform a naive pooled analysis to compute some parameters and help develop a structural model. So in this case, on the structure tab, if it's an intravenous route of administration, you can select that here in the absorption route, whether it's IV or extravascular, whether the parameterization of the model is micro, macro, or clearance, the number of compartments, and so on. And when you make these different menu selections, it actually creates what's called a model text that can be further customized if needed. For the most part, you wouldn't need to do this, but it's an option that's available if you want to further tweak your model. But typically from there, one would go on to model development, development of a population-based model. So in this case, what you could do is add some different covariates to the model. This is from the input data set, such as in this case, we have body weight and APGAR score, and those can be added to the model. And you can also identify the covariate type, whether the variable is continuous, categorical, or occasion. On the fixed effects tab, you can supply initial estimates for the parameters. And then when it comes to the model fitting, the theta worksheet will show the estimates for volume and clearance. Those are the key parameters in the study, as well as the outputs, such as the the population fit of the predicted versus observed on the data. You also get a series of residuals. These are residual plots versus time. So your goodness of fit plots, residual versus predicted, as well as ADA histograms, which will help you, tease out which covariates might be significant. as well as the population covariate plots. So you might notice a covariate that might appear to be highly correlated. For example, volume and body weight. Looks like a very strong correlation there. So you might want to run some type of a scenario where you factor in the effect that weight has on volume of distribution as part of your population model. The covariate search, so again, what I just showed you here in terms of the modeling mode, I'm in the simple modeling mode where I'm working on building my model. And then the covariate search methods can be used to help identify covariates. So in the stepwise covariate search, you can run that through and it will try adding different covariates on different model parameters and see the relative improvement of the objective function. Available objective functions include -2 log likelihood, Akaike, and Schwartz criteria. When you actually go ahead and run this, it will run a series of different combinations. So we'll try volume on weight, clearance on weight, volume on APGAR, clearance on APGAR, and different combinations. And it will actually identify which one had the best overall objective function. And you can set thresholds for what's acceptable. So in this case, it's using -2 log likelihood to evaluate whether the scenario has improved, and the significance level using p-value. So using that, this tool arrived at volume on weight and clearance on weight as the best population model. You can also do scenarios and shotgun. In the interest of time, I'm not going to go through the details of those, but just knowing that those are available as covariate search methods. You can also evaluate covariance. So covariance is if you think one or more relationships might be correlated. So on here, on the random effects tab, you can set the variance terms for the eta for volume, eta for clearance, as well as correlation between those two etas, this off-diagonal term. So you can either do diagonal or off-diagonal terms and incorporate that in the model. Next, once you have a model that looks like it's pretty good, maybe evaluate the results and see that you've got good parameter estimates. Notice that we have the effect of change in weight on change in volume. The effect of change in weight on change in clearance are now included in parameters of the model. Our coefficients of variation in this case are very good. They're all less than 20%. So one might at this point say, okay, I think I've got a good model here, but is it good enough? And move on to model validation. And that can be done by bootstrapping and visual predictive check typically. So the bootstrap is a run option mode that's available in Phoenix. So that's in this run options tab. You can specify bootstrap as well as the number of replicates you want to run in your bootstrap. And it will give you an output that shows the results of the bootstrap. So you have boot theta, and you want to see if you keep rerunning this bootstrap maybe 1,000 times, if your original model fits within the 95% confidence interval from the bootstrap that was done by resampling all of the subjects. In addition to that, there's also a visual predictive check tool. So the VPC is another tool used for model validation. It's a highly visual method that's very easy to interpret. So you see the population model and the observed and predicted quantiles of 5%, 50%, and 95%, as well as the confidence bands associated with those quantiles. So this is kind of your classic VPC, where you see the median, which is associated with the 50% quantile, and then the upper, which is the 95%, and the lower band, which is the 5%. And then from there, if you wanted to run take this model, if the VPC passes and you want to take this model and then run simulations, you could, for example, simulate different dosing regimens or simulate a different population, as long as you know that population's weight and APGAR score in this case. So this is, say, a multiple dose simulation based on the population model. Okay, so that's Phoenix NLME. I'd like to talk about one other topic as well, and that is the validation suites. So the validation suite, software validation is very important in the industry. It has been for many years in the preclinical and clinical space, and it's also starting to be more important in the pharmacometric space as well. So the regulatory agencies do require software validation to show that the software is producing consistent results in different computing environments. Historically, validation efforts might be manual test cases performed by an end user and manual cross-checking of results, maybe with a third-party application, which can take weeks or even months to complete. The WinNonlin and NLME validation suites are integrated into the Phoenix platform and provide software validation in less than 30 minutes, with the kind of the deliverable being a non-editable PDF report containing links to saved reference files, output files, and different files. So the NLME validation suite is integrated with Phoenix. It does require a software license to activate it. It runs standard test cases in the user's environment. And what it does is it runs a series of steps that's designed to test certain features of the software. So it takes a standardized input file, runs it through a standard series of steps, and produces output, and then compares that output from a similar file that was produced at Certara using the same data set and same workflow steps to achieve the result. And then it evaluates the differences between those and compares them and sees whether they're comparable or not. Okay. It also comes with the template documents in Word, such as the software requirement specification, the validation test plan, the traceability matrix that link different test cases with different functional requirements for the software, and so on. And the computational engines verification report shows comparison of results from, for example, NLME with other softwares run with a similar method. Despite all of that output that you're getting, it's very easy to use the product. You simply, with an active validation suite license, select the validation menu and select Start Validation. It runs through the different tests that test different aspects of the software, giving you a checklist as it's running to show you the status report. But typically, this is going to take on the order of minutes to complete everything. And then you get the automatic generation of the validation report, which is shown here. This is a sample snippet from part of the report. So by test case, you get a status, pass, fail, which results were shown. The results worksheet, omega, overall, or theta in these test cases, or in the case of Bootstrap, boot omega, boot theta, boot overall, and so forth. And then there's these blue embedded links that allows you to show the reference file result, the run file result, and the difference result. So, ideally, if a test case passed, that's great. But if you want to examine one in further detail, you can see the exact files that were used for comparison to produce that pass-fail result. Okay. So how are we doing on time, Sebastian?
Yes. Thank you, Chris, for the presentation so far. Great insights. You now have the opportunity to ask questions. Please use the chat pane in the control panel. And we already have some questions for you, Chris. And the first one, very straightforward. Where can I download the latest version of Phoenix?
Sure. Yeah, so Phoenix is available on the Certara website. It is here at certara.com/software/Phoenix-WinNonlin. And you simply go to this download now area, and then you can download the software. And so that's if you want to evaluate it. And then if you're interested in buying, you can contact sales to purchase a license.
Great, thank you. Second question we have here from the audience. Will the projects that Chris showed for the NCA business rules and parameter ratios be made available?
I'm sorry, I didn't catch that last part.
So I read out the question again. Will the projects that Chris showed for NCA business rules and parameter ratios be made available?
Oh, yes. They'll be made available. So I can send the example projects that I showed in this webinar to everyone who has attended here. So if you want to take a look at them and see how they work, yes, they'll be available, and we'll send those out via a download link.
Okay, great. And one more question that came through: do I need a separate license to use the validation suite you showed?
Yes, that's right. A separate license is required to run the validation suite. See in this example spreadsheet here, these are different modules that are available in the Phoenix software, and you'll get kind of this little light bulb icon showing you which licenses are active and installed. And so the different products that require licenses are WinNonlin, NLME, IVIVC, and the validation suite all require licenses.
All right. Thank you, Chris. So that was basically it. We don't have more questions.
I do have one other thing that I was asked to show, so I'll go ahead and show that and then turn it back to you, Sebastian. I apologize for that, but I did want to point out that there is an exclusive bundle offer. So this WinNonlin secured bundle includes one license for WinNonlin, a user cloud license, an admin user license, and a validation suite license. So the WinNonlin secured cloud, allowing you to store your data directly from WinNonlin via plugin into a secure validated cloud. There are top 10 pharmas and CROs using it, and the offer is valid from now until May 31st, 2022. So contact an account manager for details about this exclusive bundle offer. Okay, right. Sebastian, that was it.
That was it. Again, thank you very much, Chris. Thank you, the attendees, for being part of this webinar. Have a nice day. See you soon. Goodbye.
Thanks, everyone.